Okay, this podcast is going to pick up discussing prokaryotic cells, and in this we'll be looking at the domain Archaea, which is a unique group of prokaryotic cells. This group used to be included in with bacteria in the kingdom Monera. But with the new classification system, it was separated out and put into its own domain. So we'll talk about some of those reasons. Also in this, we'll look at a little bit about the negative and positive uses of bacteria that we didn't get to in the last podcast. But so Archaea are these unique type of extreme bacteria. They're found in these kind of extreme environments. They thrive in these extreme environments. There are a group that live in highly saline environments, and there's some that live in extreme hot environments, and there's another group that produce methane, which is really unique. But one thing that is very unique about all of these that are in this category called Archaea is that they have some unique structural characteristics. And one of those is that they have a cell wall composition that does not have a substance that all other bacteria have called peptidoglycan. So they differ from true bacteria or the domain bacteria in that means, and that's one of the major reasons why biologists thought that they should be categorized separately was for that characteristic. So before they were separated in their own domain, they were called Archaea bacteria and were a subgroup under the bacteria kingdom. Monera was the name of the kingdom. Um, so, um, so they're different in that way, but there's three different types of these, and the three types are the ones that live in high saline environments or high salt concentration are called halophils. Those that are have that ability to convert methane and carbon dioxide to methane is methanogens. And then the third group are the thermophiles, and they live in these highly extreme environments. And so this is the unique group called Archaea. Um, Now, also, we want to say something about uh, okay. Before we get to that and talk about some of the negative and positive uses of bacteria, there's some other things about this group of bacteria um, or this group called Archaea. The halophils, thermophiles, and methanogens. Is I mentioned the cell walls being different, having no peptidoglycan. And then another difference is the sequence of their genetic code. They contain some RNA, and so that sequence is different in their genetic code. And also their membrane structure is slightly different as well. So there's some other things besides the lack of peptidoglycan that warrant separating this group out into a separate kingdom or into a separate category. Now we refer to that category as a domain. So we have the halophils, thermophiles, and then the methanogens. Those are the four types of Archaea, this very unique group. Okay, the next thing we want to talk about is some use of bacteria in research and technology and some of the positive uses that we have for bacteria. We mentioned that bacteria are pathogenic and can be harmful, but we have used bacteria and prokaryotes in a number of different beneficial ways. One of those is right here in different technologies utilizing DNA that's found in prokaryotes. DNA is packaged sometimes in these circular units called plastids, and these are one of the major means of use in recombinant DNA technologies. And um, so that's a very beneficial use of these bacterial cells. Another is in a process of bioremediation. Bioremediation is a process of cleaning up the environment using living organisms. So plants and are very commonly used in bioremediation means, but also bacteria. This example right here is a bioremediation example where we have this environmentalist that's actually spraying bacteria on an oil spill. And this bacteria has some unique qualities of being able to engulf oil droplets and break those down, where normally that oil would be it would be very long lasting in the environment on this rocky shore here. But the bacteria is able to break that down so that it can degrade more rapidly. And so that's a means of using bacteria for our benefit. Another example of beneficial uses of bacteria are these little bacteria right here, B in the image. These bacteria actually synthesize a polymer, PHA, that is used to make biodegradable plastics. So it synthesizes these. Part of its metabolic processes is that it will synthesize these polymers. They can be collected and then made into plastics that are biodegradable. And so that's a benefit that we can use bacteria for. And then this last example here is the possibility of utilizing bacteria to produce an ethanol. So there's a lot of research that goes on with using this bacteria to, when it breaks down certain agricultural crops and can produce different types of alcohols or ethanols. And so that's a very beneficial use that we could have if this is used more readily and could save some fossil fuel use. Okay, before talking about a more complex type of microorganism that is eukaryotic and has internal organelles and things like that, that's going to be the topic of our next podcast. But we want to look at some different theories or scenarios about where these complex cells maybe came from. So we have our simple bacteria cells that are prokaryotic that do not have internal organelles. And so there's this theory that says that these more complex cells of protists came from the engulfing of these simpler prokaryotic cells. And that process or that theory, or it's more commonly referred to as a scenario, because it has not been supported by observable evidence in a lab, so it's generally referred to as a scenario. So here's the scenario. The idea behind this scenario is that there were these early prokaryotic non-complex cells like the cyanobacteria that we just talked about and other bacteria. And so these were engulfed by other bacterial cells, and then those cells actually ended up becoming organelles inside the cells. And so it is theorized that these could have possibly given rise to our more complex organisms that are in a category we call protists, like dinoflagellates. And we'll talk about all these in our next unit, these different types. But this little scenario is called endosymbiosis. Symbiosis is when there's two organisms that benefit each other. And so in this, there is this primary endosymbiosis where it eats one bacteria, it becomes beneficial, and then they end up engulfing other types of bacterial cells, and then they become beneficial. So it is a scenario about how possibly more complex cells came to be. Now, although these bacteria do have some similarities to some of the uh, um, larger organelles that we have, such as the two main ones would be your chloroplasts and your in plants, and then your mitochondria, the organelles that make energy in the cell. And although there are some bacteria that have some similarities to that, there's some striking differences. And so there are a lot of biologists that are skeptical about this scenario, and but it is it's a theory that is provided for the complex um, cells of eukaryotic organelles. Um, so some of the differences are some of the reasons for skepticism, and also the mechanism by which these are converted from a cell into an organelle is lacking. There's no known mechanism by how this could come about. But anyway, the next podcast we'll look at these complex organelles, and we'll look at some of the classifications of these groups. So we'll get these are much more complex than the simple bacterial cells that we've just talked about. So that'll be our next podcast.